Okay, this is our second lecture of the week for Wednesday. Now, I want to remind you of what we've looked at so far. We looked at DNA, the importance of DNA as a nucleic acid that's made up of nucleotides, and we saw that DNA is composed of these repeating subunits with a sugar and a phosphate and a base. Sugar is all the same, phosphate's all the same, bases, of which there's 4, and that's the key for variation amongst individuals. And we looked at DNA. You know, it's one thing to look at the structure, but what does DNA do? Just sits in the nucleus of our cells, and it's not really doing anything except storing information. But that information is critical to organisms being able to survive. And what we looked at so far was that DNA can be used to make more DNA. Now, we're going to look at DNA being used to make proteins. Now, that's an important function, and we've seen throughout this course what kind of things proteins do. They're involved in a whole bunch of different processes, structurally, moving things across the cell membrane, important components of cell membranes, thousands of proteins that are involved in metabolic reactions. And we looked at a lot of those in energetics. So, it's obviously important to have proteins. Where do you get the information to make the proteins that you make? Well, the answer to that is in DNA and in the form of genes in the DNA. And in order to get from DNA, where this information is stored, to a protein, there's got to be some intermediate form of information. The DNA is going to stay in the nucleus of the cell. That's a protected, special environment. DNA doesn't want to go out to ribosomes, which is where the site of protein synthesis is, and be involved in making proteins out there. DNA is going to stay in this protected environment. So what we're going to look at is RNA and the importance of RNA in acting as a go-between between DNA and proteins. So I just want to remind you again that these lectures are video lectures. It is class. It's just as much a class as if we went to the auditorium and sat there and listened to me lecture. This is the same thing, only it's a video lecture. This is material that's going to be covered on exams. You can look at these, you can use these video lectures however you want. Watching them again and again, watching them while you're laying in bed or driving or whatever. You can watch them, pause, write down notes, rewind, whatever you want to do. Okay, so if we're going to look at DNA and how you get from information that's stored in DNA to information that actually does something in the form of a protein specifically, then there's got to be some way of getting that information from DNA out to the ribosomes where proteins are made. And the answer to that is RNA. So we're going to look at RNA, and then we're going to be, after this lecture, we're going to be ready to start looking at how RNA is used to make proteins. But first we need to make some comparisons with RNA and DNA and have a good idea of what RNA does compared to what DNA does, and what the structure of RNA is compared to the structure of DNA. They're very similar in terms of structure, and there's a thought that original organisms, when they first evolved, didn't have DNA, but they had RNA, and RNA was capable of carrying out some of the same functions as DNA. Now it's a lot more complex and advanced, and organisms have DNA. So, one of the things I want to point out from the very beginning is that RNA is necessary to make proteins. In fact, I already said that, but you've got all this information in DNA. DNA isn't carrying out all these different reactions. DNA isn't moving things across the cell membranes. DNA is just storing this information. So cells, they need to make carbohydrates, they need to make proteins, they need to make nucleic acids, they need to make RNA. The cells need to make a whole bunch of different things. And in order to do that, you need proteins. So the key to information stored in DNA is that it's used to make proteins. It's used to make some other things as well, but proteins is going to be our focus. Once you make proteins, enzymes, structural proteins, whatever these proteins are being used for, then you can make carbohydrates, then you can make more nucleic acids, then you can make lipids, because these proteins, one of their most important functions is as enzymes, and those enzymes can be used to do all these other things. So the question to keep in mind this whole time when we're looking at RNA is, how do you get from DNA, which is sitting there in the nucleus saying, I'm not going out there to the ribosomes, I'm going to stay here. But at the ribosomes, proteins are being synthesized. In order to make those proteins, you need to know what the instructions are. How are you going to make these proteins? Well, you're going to make those proteins based on information in DNA. DNA is not going to go out to the ribosomes. It's not going to leave the nucleus. What's going to do that is RNA. So, what we're going to see, not yet, but soon, we're going to see that RNA is used to make proteins. RNA carries information that allows cells to put together proteins with a certain structure. And that structure is based on the sequence of amino acids with which those proteins are put together. How do you know what sequence of amino acids to put into a protein? Well, you know because that information is in a gene. Those genes are in DNA. So the whole point of this eventually what we're going to get to, protein synthesis or gene expression, is DNA being used to make RNA. DNA has very specific information in it. In fact, a gene within DNA tells a cell how to make a particular protein. But as I said, the DNA is not going to go to the site of protein synthesis. DNA is not going to go to the ribosomes. But what DNA is going to do is let itself be copied, not to make more DNA, which is what we saw with cell division, making more chromosomes. DNA is going to be used to make RNA. And this RNA is going to have the same set of instructions. It's going to be in a different format, but it's going to be the same set of instructions as in a gene in DNA that tells a cell how to make a protein. It's not in DNA anymore. It's going to be in RNA. And once that set of instructions is in RNA, then unlike DNA, which is just going to stay in the nucleus, that RNA can leave the nucleus of a cell and travel to the ribosomes and say to the ribosomes, here you go, this is how you make this particular protein that does this particular function. And then you've gotten from information in DNA to making a protein. So we're going to look at these two phases of gene expression or protein synthesis. We're going to call these protein synthesis transcription and translation. Those are to come. In order to understand how transcription works and translation works, we need to look at RNA. So what is this RNA? You know, it's RNA, DNA, you know, they've got to be similar. They're both nucleic acids, and they are pretty similar. There's a lot of similarities between RNA and DNA. So remember, DNA, we already looked at this. DNA is this double helix. It's two strands of nucleotides that are joined together at these bases, very specific order of those bases. RNA is similar in the fact that it's made up of nucleotides, repeating subunits of nucleotides. A sugar, if you look in this figure, there's a blue pentose sugar, 5-carbon sugar, a phosphate, those yellow phosphates, and a base. And we saw that in DNA there were 4 bases, an A and a T and a G and a C. In RNA, there's also 4 bases, but there's a U, uracil, instead of a T. So there's no thymine, there's no T in RNA. It's replaced by this other type of base, a U. So then the pairing becomes, the pairing is still G and C, those match up, but instead of A and T being the pair, it's A and U in RNA. Now they don't pair up with another, they don't form a double helix, but these bases can form pairs with other bases. And in this case, if you've got an A, the base that it's going to form a bond with is a U instead of a T. So what we're going to see is these RNA molecules, they're not forming bonds with other RNA molecules, they're forming bonds with DNA. The U is going to form a bond with an A in DNA. There will be an opportunity for more RNA, different types of RNA, to be forming bonds with each other based on those bases. So anyway, one of the differences between RNA and DNA is the bases. There's no T in RNA, there's a U instead. So that's one difference. Another difference is that DNA is obviously a double helix. Everybody knows that. RNA is a single strand of nucleotides, sugar, phosphate base, sugar, phosphate base, sugar, phosphate base. Some of these RNA, we'll look at these different forms of RNA. They have different shapes. Some of them form bonds with one another. It's not just this long line, depending on what type of RNA you're talking about. And then the sugar in RNA is different than DNA. It's a ribose sugar rather than a deoxyribose sugar. It's got an oxygen in it. It's just a different type of sugar. But it's a 5-carbon sugar. It's a pentose sugar, very similar to the sugar you find in DNA. But that's where the name comes from, that it's a ribose sugar. And these are ribonucleotides instead of a deoxyribose sugar and deoxyribonucleotides. So that's where the name is, a ribonucleic acid instead of deoxyribonucleic acid. And then the other thing is the lifespan of these RNA molecules. We're going to see these RNA molecules being synthesized based on the information in DNA. So some RNA is made based on information in DNA. That information is taken out of the ribosomes and used to make a protein, and then it's resynthesized, it's recycled. So unlike DNA, which is in your cells, our cells, for the lifetime of the organism, we have DNA that's the same as the original DNA we got when we were a fertilized egg. That's permanent, long-term storage of information. The lifespan of some RNA molecules is very short. You make this RNA, it's used to make a protein, and then it's all broken apart and reused again, recycled. So those are the differences between RNA and DNA. The sugars, the bases that you find in RNA versus DNA, the structure that it's a single strand for the most part as opposed to a double helix, and then the